virulence mechanism is the ability to avoid the immune response. And so one of the greatest ways that microbes do that, and obviously not all are encapsulated, but a lot of microbes that avoid the immune response do so with capsules. And so this is a classic capsule stain that you would have done in micro 224, for example, and so, or 221. And so in this particular case, you can see the halo around these little rod-shaped microbes as the capsules. These capsules are antioxidant, meaning that they don't allow phagocytic immune system cells to grab onto them and eat them. They're anti-MAC, which means that they don't allow little proteins that are produced by the immune response to punch holes in them. So MAC means membrane attack complex. And they also are often made of things that mimic host tissue, like sialic acid or hyaluronic acid, which are pretty common in animals, and us, for example. Mechanisms by which microbes avoid killing by neutrophils. So neutrophils are phagocytic cells that are pretty non-discriminating. They just eat most things, whatever, doesn't matter what it is, as long as they don't think it belongs. And so here is an example of a microbe resisting ingestion by having a capsule. So this is the anti-phagocytic thing that I'd mentioned. And there's a variety of reasons why that happens. Sometimes the receptors on the microbe that the phagocytic cell would be looking for are covered by this slime layer, this capsule layer. That's one example. And that's a big reason why that happens. Sometimes microbes just outright kill the neutrophil, so they'll produce a toxin that destroys the neutrophil before it can eat it. And those are pretty deadly. Well, they're all deadly, but microbes that grow inside phagocytic cells, they simply just let themselves be eaten, but then they can grow inside the phagocytic cell. And so I'd already mentioned Salmonella as an example of that, using its various type 3 secretion systems to avoid being destroyed. And so then the bacteria can then break out of the vacuole and then grow in the cytoplasm if necessary. Some of them prevent phagolysosome formation. So if my pointer is a lysosome rather, it'll combine with the phagosome to release toxic chemicals in there to kill things. And so some of these guys can just prevent that binding from taking place. So I showed you that with Salmonella. Some just escape the phagolysosome before the detoxification happens. So if this is the phagolysosome coming over, these guys just bail and they're not present when the lysosomal fusion takes place. And some of them, they don't even care that the reaction happens, and then the microbes just kind of deal with the onslaught of toxins. Of course, the antibody response is really important, and so the microbe, I've already mentioned this, but the microbes, if they have fimbria, for example, or capsules, the microbes can change the protein structure of these and confuse the immune response. So I've already mentioned that. But some microbes just outright degrade antibodies. So IgA is an antibody that's often produced to protect the mucous membranes. And so Neisseria, being an organism that colonizes mucous membranes, particularly the urethra, will produce a protease that breaks down IgA. So here's an IgA molecule. And each of these little regions right here is an antigen binding site. But this microbe will produce a protease that can chop this all up so that it can't work. So if we produce this antibody to attack this microbe can counterpunch by chopping that up. Some of the types of exotoxins that microbes produce fall into different categories. And it's the type 1, 2, and 3s. The other type are the endotoxins, which I'll talk about as well. But type 1s are the superantigens, as the name suggests. They sort of overstimulate the immune response. And type 2s are toxins that damage eukaryotic membranes. And third are the AB type toxins, the type 3s. As far as the others go, endotoxins are part of the gram-negative outer membrane. And so the basic structure is a lipid followed by a polysaccharide. More specifically, though, the lipid is called lipid A, and so it's this molecule here. And so this would be embedded in the outer surface of the gram-negative bacterium. And so then there's a fatty acid layer, And so again, this makes up the outer membrane of the bacterium. And then sticking out into the world is a core of sugars. Each of these little patterns is a sugar, different sugar molecule, like hexoses, for example, with different decorations. And then, so this is a whole core that's close to the cell surface of sugars, and then an O-specific chain that sticks out into the world as a little stream of sugars. And so, if the microbe were to be, for example, lysed because of an antibiotic that you're taking, and this portion of that microbe releases into the bloodstream, that could make you sick and actually kill you. And that's sometimes why individuals die from gram-negative septicemia type infections. They are given antibiotics. The antibiotic may work really well and kill the microbe, but the microbe releases this compound here that you see, and that causes a wave of problems that are illustrated here. So imagine bacterial lysis took place. Again, this is gram-negative bacteria. So the gram-negative bacteria fall apart and their LPS is released into the bloodstream or wherever. We produce an LPS binding protein, which will bind to those LPS pieces. And then that triggers macrophages with this particular CD14 receptor to bind to them. And that combination triggers these guys to produce a whole bunch of cytokines, immunomodulatory chemicals. Some of these same chemicals, like IL-6 and tumor necrosis factor, are the reason why COVID-19 disease is very dangerous for certain individuals. The overproduction of these cytokines and, again, immunomodulators does all these bad things, activates the complement cascade. And one of the things the complement cascade does is causes coagulation of the blood. These guys will also trigger endothelial damage, and endothelial cells are cells that line the veins and arteries. And so, again, activating the coagulation cascade can block small capillaries and prevent blood from getting into places, and tissue can die. So you could, again, mini strokes and things like that can happen in these particular cases. So again, the bottom line is all this stuff results in endothelial cell damage and multiple organ system failures. And so again, endotoxin in and of itself, just pieces of the microbe, can cause severe disease. So one of the types of toxins I should back up and say that these three types of toxins are protein toxins. However, endotoxin is not a protein. It's lipopolysaccharide. So again, these are proteins. This is lipopolysaccharide, so a combination of lipids and sugars. That's what this is, lipids and sugars. So these are the type 1 protein toxins, and an example of that is toxic shock toxin that's produced by staph and strep species. And so these organisms would produce these toxins referred to as superantigens, and these superantigens will cause antigen-presenting cells, or APCs, like macrophages and so forth, to have a weird interaction with the T cells. Normally, these guys interact, in a very controlled way, and so that when this guy recognizes something bad is infecting the host and kills it, it can then show that to the T cell, and then the T cell can understand what the infection is and then properly proliferate. However, there's an unusual binding that takes place here between the superantigen and the macrophage. Between the T cell, the superantigen, and the macrophage. And as a result, the superantigen stimulates far more T cell replication. So excess T cell replication results in a cytokine storm. And again, you keep hearing cytokine storm with COVID-19, but that also happens with other organisms like staph and strep. And so that the net result of that is shock. And again, when I refer to shock, I'm talking about all the things that I mentioned here, the coagulation cascades, the endothelial damage, and so forth. So in this particular case, the type 2s, these are the membrane disrupting toxins. And so they can be large pore formers, as pictured here. So the bacterium that causes strep throat, or the bacterium that causes listeriosis, or the name Listeria, produce pore-forming toxins. So some of the strep throat problems that you have when you get strep throat are caused by a pore-forming toxin called streptolysin. So here's our pore-forming toxin, and it finds different receptors in the cell membranes that it's looking for. So once these guys bind and find the receptor, they can polymerize. Additional pore-forming toxin monomers can bind and form full-on pores. And then these pores cause cell leakage. So lots of things can spill out of the cytoplasm and kill the cell. Another type of membrane disrupting toxin are the so-called phospholipases. And so these guys will chop the little hydrophilic heads off of these phospholipids and cause membranes to fall apart. So again, phospholipases remove polar head groups from lipids and cause the membranes to fall apart. The type 3 toxins are the AB type toxins. As the name suggests, they have an A portion and a B portion. And so the B portion is for binding. The B portion binds to whatever cell it is, and then the A portion goes inside and does some kind of terrible toxic thing.